Hello everyone, welcome to Science and Tech series with Dr. Vivek Rana. Hope all of you are doing well. Topic for today's discussion is from Biotech Medicine section. We'll be taking up important milestone in Indian biotech sector, development of India's first ever indigenously developed mRNA vaccines against Omicron variant. So the vaccine has been termed GEMCOVAC-OM. So we'll be discussing in this theme what is the overall significance of development of this yet another vaccine in the COVID-19 arsenal, which is going to help us tide over in case of any future eventualities of this, you could say the virulent strain of neo-cov SARS. Now, to understand significance of this topic, we will have to go a little back how vaccines eventually developed in case of COVID-19 vaccine in wake of that global outcry when we have seen the first wave and the second wave almost going out of hand of most of the developing as well as the developed world. And after the sanitizer and all those, you could say, basic isolation protocols and lockdowns, only vaccines provided the breather to humanity. And the first ever vaccine that was approved or developed was Sputnik V. The Sputnik V was a Russian, you could say, first ever vaccine, and it was named after the first ever satellite launched by Russia, which heralded a space race between, you could say, that Cold War nemesis US and Russia. And in similar way, the first vaccine triggered a similar kind of, you could say, race in development of different types of, you could say, the vaccine variants. So India was not far behind, and it soon developed its first ever indigenously developed vaccine, which we all know is Covaxin. At the same time, the Pfizer, Moderna developed a new type of vaccines, which are mRNA vaccines, which were, you can say, first time they were used on a large scale for any human disease, even though some isolated examples beforehand have been in picture. But the Oxford AstraZeneca vaccine Covishield dominated the headlines, and in India it was Serum Institute of India which took the burden of manufacturing the Covishield variant in India. So basically, when we are talking about the significance, we need to understand how different vaccines work and what are the different types of vaccines which are currently available. So even in UPSC mains in 2022, there was a question on how what are the different types of vaccines available, how their mechanisms differ from one another. So basically, the Indian vaccine Covaxin was more or less an inactivated vaccine. In simple terms, we can say the dead part of the, you could say, the variant of virus is introduced, and we are trying to make the response from antibodies against these spike proteins. So it is comparatively a safer mode to administer because rarely there would be exaggerated immune response against a dead, you could say, virulent strain. So this is tried and tested method used in lot of vaccines development, where we just introduce a part of virus or a dead part of virus to elicit immune response. Now in Covishield, we is considered more effective than Covaxin because in terms of, you could say, the immune response, because in those we use the viral vector vaccine. Adenovirus, which commonly causes common cold, is weakened and introduced in body to once again trigger antibody response, so that whenever the real virulent strain enters the body, the active form of the antibodies can neutralize them. So basically in Covishield, we use a live attenuated vaccine, which are quite effective. Now, generally, these were the two basic protocols which are generally used to counter any emergency, you could say, virulent attack. But in USA, we have seen Pfizer and Moderna developing a new type of vaccines called mRNA vaccines. Now, this M in RNA stands for messenger, and RNA is ribonucleic acid, because we all know the COVID-19 virus was a RNA virus. So, in that case, the word mRNA comes into the picture. Now we know the RNAs, or you could say the ribosomes. When we study, the main function of ribosome is to produce proteins, and from proteins we can produce antibodies as well. And we have to understand out of MT and rRNA, messenger RNA is the main RNA present in nucleus, which triggers formation of proteins via ribosomes. So this technology doesn't introduce any part of virus whatsoever when generating an immune response. So in a way, we can say these are comparatively more safer than both the inactivated as well as the live attenuated vaccines. The only drawback of these mRNA vaccines, which were developed by USA companies, was they required extremely low temperature for, you could say, for refrigeration, because mRNA being fragile molecule, you could say, easily gets denatured. So India, even though had an option to acquire these vaccines as well, but we went for Oxford AstraZeneca Covishield because we didn't have adequate cold storage facilities. And even though we were also skeptic about how this vaccine is going to evolve, because all countries were going hit and miss trial, so eventually it was a good decision that we opted for Covaxin, Covishield, Sputnik, and other variants eventually. So basically, we have to understand that only reason why we couldn't afford, or you could say acquire mRNA vaccine, was the problems in the cold storage facilities. So the current vaccine, now coming to the main theme, because now you have got the adequate background, what are the different types of vaccine, how they eventually work. So in current case, when this project was supported by Department of Biotechnology and BIRAC, we have developed this model successfully. And this mRNA vaccine has, you could say, two major advantages over the conventional vaccines developed earlier. The first one is that it doesn't require extreme cold storage facilities. It can be easily maintained in the ordinary refrigeration facilities, which India already has. We can recollect in polio vaccine as well as in other, you could say, similar vaccines, we use the common cold refrigeration procedure. So this is adaptable to that. So in a way, we have overcome a major hurdle of preserving mRNA vaccine in Indian conditions by development of this basic vaccine. The second major advantage is that now we have seen the Covaxin, Covishield delivering results, but in third wave we have seen lot of mutations occurring in the original SARS neocov virus strain, and lot of variants of concerns emerging, right from alpha, beta, gamma, delta, and finally it was Omicron, which was creating havoc in China and other parts. Even though in India the delta strain was, and Omicron had also a nightmarish experience for a lot of individuals. So as a booster dose, this vaccine could be quite effective, because this has been designed specifically against the variant of concern, Omicron strain. So we can say that those initial vaccines which we used for as a primary vaccines, they were primarily effective against the original strain or the minor variants. For Omicron strain, we have seen the response being less than optimum. So it was a lot better to back it up with more effective vaccine like GEMCOVAC, which is designed specifically for this purpose. So you can say that it's quite a milestone that for even a variant we have developed a vaccine. Now, another major advantage of this particular vaccine is the mode of administration. We have seen that in second wave, after the first dose, lot of you could say people didn't go for the completion of the course because of, you could say, vaccine hesitancy. Now, vaccine hesitancy could be due to some rumor mongering that it is likely to trigger some hyperactive response, or may result in death due to isolate one or two example which are at times in media, or at times it is mainly due to needle phobia, because in childhood you all have some unpleasant memories of doctor administering a needle and resulting into some painful experience. So a lot of people didn't want to relive or go through that agony again and again in form of booster dose, or you could say the second dose. So currently we have seen the evolution of intranasal vaccines, and in this case there is no need to go for that same needle, you could say, injections. It is designed to be administered through the pharma jet system, so comparatively less traumatic, or even you could say painless procedure, which is likely to result in more compliance than the one which we have seen earlier. Even though India has done reasonably well and currently we have tided the wave successfully without any scare, because the vaccine coverage was quite adequate, and we backed it up with Arogya Setu and other, you could say, application through which the people have resolved to fight this pandemic with much more resilience. And currently we can say India can say that it has handled the COVID-19 pandemic in a much matured way than anticipated in long run. So this is just one of the more stepping stone, and in future, if such alarming emergencies arise further, so we can back ourselves that our biotech sector is ready to evolve any new technology which at times may appear to be difficult to emulate, but we are delivering the indigenous version. So this topic holds specific significance not because we are going through some emergency, but because we are now proactive in developing our responses to future exigencies. So there's a syllabus theme which is formally mentioned in UPSC, indigenization of technology. Even though the full-fledged question might not get repeated about the vaccines, but still you can quote this example that how India has developed a key technology, and it's the milestone in development of a new form of vaccines may be helpful in further case or developing more such themes for other, you could say, infectious diseases as well. So make sure you are clear about what mRNA is, what are different forms of RNA, what are protein factories, how central dogma works from DNA to RNA to ribosomes, and apart from that, the basic factual information, which institutions developed which variant, what are the technologies behind them, and what are their efficacies level. So even in state exams, we can anticipate one question from this section once again. So make sure you are brush aside with all the relevant facts. Apart from that typical theme of COVID-19 disease in news, the system affected, the, you could say, the RT-PCR test being the gold standard, the nature of communicable by means of droplet airborne infection, as well as the various measures which we use. So if you have any specific doubt which you feel that it's not been addressed, you can ask them in comment section. We will try to revert with genuine your queries as well. So this is all what we have in today's science and tech section on biotechnology. Another major milestone. We'll be back soon with another current affair on science on environment next week. Thank you for joining. Have a nice day.